Good afternoon. Thank you for joining us live at NASA's Kennedy Space Station here in Florida. I'm Marie Lewis with NASA Communications. We're here to talk about what's on board for SpaceX's 14th commercial services resupply services mission to the International Space Station. Launch is targeted for Monday, April 2nd, tomorrow at 4:30 p.m. Eastern Time from Space Launch Complex 40 at Cape Canaveral Air Force Station. Our guest today will talk about some of the research that makes up the 5,800 pounds of cargo packed inside SpaceX's Dragon spacecraft. We will call up our guests individually to share more with you and give you each a chance to ask questions as well. If you're following us online, you can send in your questions on Twitter using hashtag AskNASA. Our first speakers today will give you an overview of the research on board. We have Pete Hasbrook, Associate Program Scientist for the International Space Station Program at Johnson Space Center. Dr. Craig Kundrat, Director of Space Life and Physical Sciences and Applications Division at NASA Headquarters. And Patrick O'Neill, Marketing and Communications Manager for CASIS. Gentlemen, thank you.
Thank you, Marie. Thank you all for being here today, and we'd like to thank all of our friends and fans who are dialed in and watching us on the internet. It's always great to be here to give these kind of overviews of the science that we're doing on our commercial resupply services missions, this one being the 14th for SpaceX. The ISS Laboratory is what I'm here to really talk about, and it's a multidisciplinary, unique laboratory in space, a world-class laboratory. And our commercial providers, not just the cargo vehicles, but all of the hardware providers, do a lot to keep the science moving for us on the ISS and returning benefits to all of us here on the ground. This SpaceX mission, as I think you've heard, is launching almost 3 tons worth of science hardware and samples for us for the space station, and that includes the external experiments that will be brought up and put outside the space station. Over the course of our current increment 55 and 56 mission, we have somewhere over 280 different experiments going on. This SpaceX mission is going to help the progress of at least 50 of those experiments, so it's a big chunk of the work that we're doing on station. And over the course of the history of the ISS, and by the way, we are celebrating our 20th anniversary of the ISS coming November, the anniversary of the first Element launch. We've had over 2,500 different experiments on the station, over 3,000 different principal investigators represented, and somewhere over 1,500 peer-reviewed scientific publications that we've been able to track. And those are important to us because that shows how the results of the space station are being disseminated to the rest of the world, to the science community, and to the rest of us. I hope you don't mind me using notes. There's a lot going on and there's a lot to keep track of. In the world of space biology, we've got a couple of cool experiments. We've got one that's studying the grass Brachypodium distachyon, which is also known as commonly as purple false brome. It's a grass that scientists are studying, and they're going to compare it to the performance of Arabidopsis thaliana, or thailcress, which has been studied for years on space station, but now they'll be able to extrapolate Arabidopsis over to the false brome and learn more about how their grass might perform. We have a couple of experiments going on in the area of fruit flies, and fruit flies always amaze me how they can teach us so much about ourselves, because fruit flies' genetics are remarkably similar to humans' genetics. Oh, there's a phone that somebody forgot to silence. Remarkably similar to ours, and we can learn a lot about, for example, our autoimmune system and how humans might respond to diseases. There are a couple of different fruit flies missions or experiments launching that you'll hear about a little bit more. We also have things going on in the plant research area. An experiment that's been going on is the plant gravity perception, how plants use gravity to know which way to grow and how to grow efficiently. And you'll also see and hear about some hardware that we're launching to help improve our watering and our nutrient delivery system for our facilities on station. There's a lot going on in the area of human research. SpaceX isn't really necessarily bringing up a lot to the station, but I want to tell you what's going on. We're involved in an experiment called marrow, which is studying bone marrow, the marrow in our bones that creates fat cells as well as red blood cells, and it's studying to see whether the creation of fat cells is happening at the expense of creating red blood cells. There's an experiment called cerebral autoregulation, which is studying our brain's ability to regulate blood flow even when it's maybe deficient from the heart and our blood vessels. We're continuing fluid shifts, which is an international experiment that studies the effects of weightlessness and microgravity on astronauts' vision and the effects that are seen that are detrimental there. We're studying bone turnover improvements and how the bones regenerate themselves and how we might use that to learn about age-related bone density issues here on the ground. We have some technology demonstrations that help us in the human science world. We have hardware that'll study cardiopulmonary systems without restricting astronauts' mobility. We also have advanced clothing studies, which will help astronauts keep cool and to wear their clothing well when they're exercising on space station. All of these experiments that I've just talked about have the benefit of being internationally related. They either have international scientists as the principal investigators, or we are leading them and we have multilateral participation from the scientists. All of them use our international crew as subjects. In the technology demonstration area, we have several things going on with technology, especially advanced computer systems, how they work in the harsh environment of space, multi-platform processing. In physical sciences, we're launching a sample container that will study sintering, which is a physical process creating solids, creating metals through just heat and pressure without melting the powders that they're using. We are continuing to study containerless processing of metals and glass and alloys through the electromagnetic levitation furnace. We also have the external facility, the MISSI, or the Materials for ISS experiment, the flight facility. It's a good example of the partnership between the National Laboratory, because it's a commercially provided facility, and NASA experiments are going up with the benefit of being the validation experiments, the first experiments to show that the facility works. Pardon the sticky card here. And finally, the SpaceX has a great benefit for us by being able to return cargo and samples, especially in the life sciences area, whether it's cell research or human research. The plant and the fruit fly samples all come home in the SpaceX. It also brings hardware home for us so we can refurbish it, see how the technology demonstration hardware worked. An example of a big piece of gear that we're bringing home is the Robonaut. Robonaut's had some issues over the last couple of years, and we're finally bringing it home. The engineers believe they know how to fix it, and it'll be available to launch again. One more thing that I want to tell you before I cede the microphone is our Program Scientist Toolbox. I'm in the Program Science office, and we maintain the database of all the information about these experiments, everything I've talked about and more. We've worked over the last year to improve that interface out there so you guys can search better. And we just put out a release this last week that improves the search capability, so you'll be able to bring things up a lot more easily. You can find that by just doing a web search of Space Station Research Explorer. You can go to www.nasa.gov/station experiments, and you can browse from there. And if any of you are going to bring it up right now, and you can on a smartphone, I'll just advise you to turn it sideways and the display will work a little better. So that's a quick overview of a lot of the science going on, and I'm happy to turn the microphone over now to our good friend, Greg Kundrat. Greg, thanks.
Thank you, Pete. And thank you all for coming out today, and for those of you joining us online. I'd like to take off from where Pete left off and go into a little bit more detail on 3 investigations in the space life and physical sciences realm at NASA. The mission of our division is twofold: to enable exploration and to pioneer scientific discovery for benefits back on Earth. And all 3 of these investigations support both of those missions, as you'll see in a moment. So the first is Apex 6. Pete mentioned Brachipodium. This is a workhorse model organism in biology. You may recall from your high school biology, there are monocots and dicots. We have been doing a lot with dicots, like veggie and the lettuce, Arabidopsis, another workhorse. We haven't done so much with monocots, and so this is really a pioneering experiment for us. The investigators will be looking at the seeds germinate and how the roots grow in microgravity. They'll be taking video during the growth on station, then harvesting the tissue and doing genetic expression analysis back on Earth. And that data will also go into NASA's Gene Lab project, which will make the data available to the wide community so that other investigators can compare the results of Brachipodium to their own systems of interest. Apex 6 is going up on SpaceX 14. Coming down, on the ride down, plant gravity perception, which Pete also mentioned, is studying that workhorse Arabidopsis, a dicot. That experiment carefully looked at how Arabidopsis was growing as a function of simulated gravity. So on the space station, there was a centrifuge that was spinning and had samples of seeds at different radii, so they would get different levels of gravity ranging from 1 G like we have here on Earth down to 6,000ths of a G. And so they were looking at how the seedlings were growing in response to those different G levels. Not only that, they had normal Arabidopsis, but they also had a mutant which doesn't make starch and takes away the normal mechanism that those plants use for sensing gravity, which is how starch particles sediment within cells. So they're looking at see what kind of pathways are used when you don't have gravity or when you don't have those starch particles. Like Apex 6, it's going to, it has video of the on-flight phase of the experiment, and will do extensive gene expression analysis back on the ground. Now, both of these help us in enabling exploration in that we are learning how to grow plants in space for exploration, be it for food or for life support or some combination of the two. It's also beneficial for life back on Earth because it turns out that how roots grow and their architecture is a major determinant in how resilient they are. So we might be able to design more resilient crops if we understand the mechanisms at work and can improve the root structure. Now for something quite different, we switch to material science and the sintering experiment that Pete mentioned. That is looking at tungsten alloys, metallic powders, which when you heat them up, without liquefying them, it turns them into a solid. The process is very much like taking a snowball, from those of you up north, and packing it really hard. And so you get that ice ball when you throw it, it really makes an impact. Very similar kind of process. You don't melt the whole thing, but rather the temperature binds the particles together. It's a $25 billion industry in our country, and it's used to make things from metal cutting tools to self-lubricating bearings. And it turns out in microgravity, the particles aren't sedimenting like they do on the ground. And so that means that pores form more readily, and it also turns out that the final product can be more distorted internally. And so that's a challenge for microgravity and exploration, because if we want to use this kind of technique to make fresh parts just in time, on demand, in a deep space mission to repair equipment, we have to work out how this technique works. It's also an area that's only been studied scientifically for about 50 years. And so there's a lot to be learned. And so looking at these alloys that are made in space, comparing them to ones that are made on the ground by exactly the same technique, except it's in 1G, will allow investigators to sort out the component, the role of gravity and how that affects the final structure. So it'll arm us for information for enabling exploration and may also help us improve some ground processes as well. So that's kind of a recap of going a little bit deeper into 3 investigations within the space, life, and physical sciences realm that are both enabling exploration and pioneering scientific discovery. And for more information on investigations that benefit life on Earth, I'll turn you over to Patrick and Cases.
Thank you very much. And happy Sunday, everyone, and thank you very much for coming on out and joining us for this launch. So a little bit about the National Laboratory. So the Center for the Advancement of Science and Space, otherwise known as CASIS, we are tasked with managing the U.S. National Laboratory on the International Space Station. A lot of people might not have known that the space station actually is a national laboratory, and a little bit of background on that. In 2005, Congress looked at the research that was taking place on the space station and said, man, NASA's doing some terrific things relative to living in space, better understanding the human condition so that we can be in a microgravity environment for extended periods of time, developing technologies that will allow for us to maybe one day go beyond low Earth orbit. But there's a lot of other things that are happening on the space station that maybe we could take advantage of, whether that be from a commercial company or an academic researcher, an innovative startup, another government organization. What if we were to open up the access and the opportunity to all of these entities to be able to take advantage of this incredible research facility that we now have? And so that's what ended up happening in 2005. They declared the U.S. portion of the International Space Station a national laboratory. And in 2011, CASIS was ultimately selected by NASA to manage the U.S. National Laboratory. So we get up to 50% of the research that goes to the space station, 50% of the research that goes down, and up to 50% of the astronaut allocation dedicated towards that experimentation. So we really do have an opportunity to partner heavily with NASA on all things that are happening on the space station to utilize it for the greatest capacity to benefit life on Earth as well as our exploratory endeavors. So switching into 2018, it's always exciting to come to a rocket launch because first and foremost, it's the smoke and fire. But on top of that too, it's also really an exciting time because we're starting to enter what I like to call the golden era or the golden years of research on the space station. In 2017, the National Laboratory had its best year yet as far as investigations going to and from the space station. In 2018, we're going to be building on that. And, when I say that it's kind of the golden years, it's because there was a build-up phase of letting people know that first and foremost, there is a space station. Secondary to that, too, it's a great research facility. You can do things that you can't do or can't mimic here on Earth. And lastly, especially when you're talking to commercial companies, it's nowhere near as expensive to take advantage of this platform as you would think it to be. And you can benefit from it from a branding, marketing perspective. So, again, it's been a process over the last few years at CASIS, and we're really excited about where research is going to be going. And so, I think now segueing into this particular launch. So what's going up on the ISS or the ISS National Lab on this particular mission? So, we really are running the full gamut of research from life science investigations to companies and other researchers who are sending their continuation payloads to station, which is another important factor to look at when you're using the space station. Is it's not looking at college basketball one and dones. You're looking to have people want to send a series or a fleet of investigations continuously using the microgravity environment that the space station is able to provide you, because hey, at the end of the day, it might have far-reaching benefits to your research or your product here on the ground. One thing that, and Pete talked about it from a life science standpoint, we see it very heavily with SpaceX in particular because of their return capability, where you see an awful lot of life science investigations that go to the National Lab. And it's a terrific thing because on this mission, the same thing can be said, where we have everything from protein crystallography experiments to student investigations looking at DNA, to looking at just all sorts of disciplines, plant biology research. But then the great thing too is that we also have our partners at Orbital ATK. And they oftentimes will see a much more slanted emphasis on material science and physical sciences. So here with SpaceX, you see a heavy push towards life sciences for the National Laboratory, and our other partners at Orbital ATK are kind of helping to really close off that continuum. And that's the best thing about the National Lab, again, is that all types of scientific research can be done on this facility. And on top of that, the facility is constantly adapting. It is constantly molding to the needs of what the research community is looking for. A little bit later on today, we'll be hearing from TechShot, who's going to be talking about a new facility that is going to be joining the space station that's going to be enabling life science opportunities down the road. And, what we're trying to do is not only have a unique platform, but a world-class platform for innovation. And the more facilities that we have that cater to the research community, the greater capacity we're going to have for sending more research to station, and then ultimately the greater capacity we're going to have for the types of discoveries that can improve or change the way that we live our lives here on Earth. So that's kind of the National Lab 101, and I think at this point, are we going to be answering any questions?
Yes, so we do have time for a question. So if you have a question in the audience, if you could just raise your hand so we can get a microphone over to you, and then please direct your question to Pete, Craig, or Patrick.
Hi, Chris Gephardt with NASA Spaceflight for Pete and Craig. You mentioned Robonauts coming back after about 7 years. Can you talk about what you saw that went wrong that you want to bring Robonaut back for? And will another one go up based on how helpful Robonaut 1, let's say, was for the crew? And then I don't remember high school biology and botany as well as I should. Can you explain some of the other terms that you used and how they differ from the other growth experiments that have been up there since? Thank you.
Cool. I wanted to know what a monocot and a dicot were myself. Answering the Robonaut question, the engineers have looked at why Robonaut wasn't able to power up on board. They had the benefit of being able to have the crew do a lot of troubleshooting, which is an advantage of having a crew in space. If something's not working, you can have your crew dig into it and see what was wrong. And through the crew's help and through a review of a lot of imagery and comparing to what's on the ground, the other Robonaut units on the ground, They've figured out that they believe there's something in the electrical system, some kind of a short, and I think it's related to something that is not there in the configuration of the Robonaut that's on board that is in all the subsequent ones on the ground. So they are pretty confident that when they get it back and they dig into it, they'll be able to repair it fairly quickly. I believe they are funded and have a plan to launch it again on an upcoming commercial resupply vehicle, maybe a little over a year from now. But they are confident that they'd be able to take this one and repair it and relaunch it. Craig's question.
On the monocot dicot distinction. So if you open up a seed with a monocot, you see one like vein. With a dicot, you see two. When you look at roots, the monocot's kind of all over the place, kind of a fibrous tree, whereas the dicot has like a tap root. If you cut open a stem, the monocot's kind of got fibers going up all over the place. In a dicot, they're well organized. Probably the easiest thing, if in springtime like now, if I remember right, the monocot has flower petals in multiples of 3, and the dicot multiples of 4 or 5. So there are two major branches, and the underlying biology, differs at a significant level.
Okay, and right here?
WSGU radio. The Apex 6, how long do you intend to have it in orbit as far as to get the results back you're looking for?
I think the growth phase, so Howard Levine, can you help me out there? It's only a matter of days.
Right, it's actually a 5-day experiment.
Yeah.
Let's get a microphone over to you.
What?
So it's actually a 5-day experiment, and then the plants will be fixed on orbit and put into Melfi at -95, and then brought back when the Dragon returns. By the way, that was an excellent answer by Craig on monocots and dicots. A simpler answer is that the higher plants are split up into 2 subclasses, monocots and dicots.
Okay, thank you. Yes, right here in the front. Let's get a microphone to you first.
Hi, Ken Kramer, Rocket STEM and Space Up Close. Probably for Pete. Can you talk about Congress and the President just passed and signed this omnibus budget? How did the ISS fare in that budget? And maybe Pat can talk about it too. And the other thing I want to ask about is late load items. Are there any late load items, and can you talk about what they are?
I honestly can't say too much about the budget. I just am not close to that in the job that I do. So let me pass that, and maybe somebody else will have a better answer for that one. Late load items, you'll see several. There are plant experiments that are launching late. A lot of the cell biology is loaded late into the capsule. I'm having a little trouble with some of the other ones. I'm not so familiar, but that's been a big campaign. I know you may have a chance to meet some of our social media folks who are here from the Houston office, who are getting a great opportunity to go around with the folks that are doing the late load and able to video some of it, and that'll be coming out on social media. So can't answer very well for you, Ken, but at least I can plug my friends who can answer that. So follow ISS_research and you'll see the results.
I don't want to cut this short. I think there were a few more questions, but we are running a little tight on time, so I want to introduce our next speaker. Thank you, gentlemen, very much. Next we have Dr. Dan Close. He is the, oh, go ahead, please. Sorry. Dan Close is the Chief Scientific Officer at 490 Biotech, and he will talk about the company's metabolic tracking investigation into the impacts of pharmaceutical drugs. Dan?
Thank you very much. So just some real quick background about 490 Biotech. We are a startup level biotechnology company based out of Knoxville, Tennessee. So we're one of those people that's very proud to be able to take advantage of the International Space Station National Lab and use that to try and help improve life on Earth. We do this because our company is developing the next generation of bioluminescent imaging technologies, which of course begs the question, what is bioluminescence? And that's just a fancy scientific way of saying the production of light from a living organism. So when most people think about this, you can think about fireflies blinking in a field in the middle of summer in an idyllic setting. But unfortunately, the current iteration of bioluminescent technologies, just like the fireflies, are sort of one and done. They blink, you get a little bit of information, and then you have to start all over again. So what our company does is we've developed a new technology that we call autobioluminescence. And this is basically a big step forward in bioluminescent imaging, where instead of intermittent snapshots of data acquisition, we can continuously monitor any living cell, whether that's a bacterium, whether that's a yeast, whether that's a human cell culture or a small animal model. And we can track their metabolic activity in real life. So what does this actually look like? I did bring a picture with me today. So we do have some cells of ours growing in a dish, and you can see very clearly which wells contain living cells and are helping us out with our self-promotion today, and which cells are empty. So we look at these and we actually use this light to judge the health of a cell. So healthy, happy cells glow very brightly. As the cells become sick, they become dim, and if they die, the light goes out. So why is this important? On Earth, we've been using this with pharmaceutical companies in order to reduce the cost of new therapeutic development. So they can use this technology to watch these cells in real time and monitor the effects of potential new therapeutic compounds. So they can tell if those compounds are beneficial, if they're making cells healthier, happier, and brighter, or if they're detrimental. They're taking healthy tissues and they're making them dim. So our experiment on board the International Space Station is to take advantage of something that's been known for a long time but has been very difficult to leverage from a scientific standpoint. And that is the somewhat ironic fact that when you grow these types of human cells in a dish, if you grow them in microgravity, their physiology, the way they behave, is actually more similar to how they behave in the body than if you grow them in a dish on Earth. And that's because when you take them out of the body and subject them to gravity within a dish, they all form a single layer on the bottom. And because we are all not two-dimensional, it's very easy to see that that's not their preferred way of growth. When we grow these cells on board the International Space Station, however, without gravity to push them to the bottom of the well, they naturally form three-dimensional structures. The same types of three-dimensional structures that a lot of companies now are spending lots of money and lots of exotic resources on trying to encourage here in normal gravity. And those three-dimensional structures that are imparted by these strange technologies that are being used now on Earth can have adverse effects on the way that these cells grow, which in turn makes it more difficult to predict the efficacy of those potential new therapeutic compounds. So our experiment will actually be taking these glowing cells, traveling them up to the International Space Station, and then performing parallel experiments. One on board the International Space Station, where we'll be treating these cells with known therapeutic compounds, and one in parallel on Earth, we'll be treating them identically, but using regular Earth gravity. We will then be able to demonstrate the difference in the effectiveness of these therapeutic compounds between low Earth orbit and down on Earth by using a combination of compounds that were eventually approved for usage within human subjects and those that eventually failed at the clinical testing level. Our hope is that we can demonstrate that by taking advantage of the more natural human physiology that occurs within these cells during growth on the International Space Station, that we can provide a way for these pharmaceutical companies to do their screening at low cost in low Earth orbit and get better information about the safety and potential functionality of these new drug compounds. If we can make even a minor dent in the number of compounds that inappropriately pass phase 1 testing and fail during much more expensive phase 2 or clinical level testing, it can more than pay for the cost of launching these compounds into space in order to do these evaluations. In addition to doing this, we have a second experiment where we'll be doing the testing up on the International Space Station, and we'll actually be returning the collected cells to Earth to evaluate the ability to do processing after return from the station. So our hope is that we can come up with an almost fully automated system where our technology, these cells under study, are sent up to the International Space Station along with the therapeutic compounds. Then they can be tested, the data can be beamed right back to Earth, or if that doesn't work out, we can take the cells back and I would be more than happy to test them in my lab, as long as you don't have too many of them. So I think it's a very interesting project and something that could have some far-reaching effects if we're able to demonstrate what we believe we'll be able to demonstrate, and hopefully we'll be able to do a lot of good for a lot of people. Thank you.
All right. Do we have any questions for Dan? Yes, here in the front.
Yeah, Ken Kramer, Space Up Close. Can you describe the hardware a little bit? How are you going to run this experiment? How big is it? How heavy is it? Thanks.
So that's one of the main benefits of our technology relative to the existing technology is you can imagine if you had thousands of compounds that you wanted to test, that's hundreds of thousands of samples because you can only look at each one once. Because we switched from intermittent observation to continuous observation, we can condense all those down to just a single replicate sample. So depending on the number of compounds that are involved, we're doing a very limited study. We're looking at 5 compounds initially because we, of course, for demonstration purposes, wanted to cherry pick those compounds that we knew had either successfully gone through trials or had eventually failed in clinical trials. But the amount of savings would be multiplexed over the total number of total samples that were tested. In terms of equipment, we're using all equipment that's currently present on the International Space Station. We're not requiring any new technology to be flown up to the International Space Station in order to do this experiment. Thankfully, it's actually a fairly simple experimental design. Our cells are pre-packaged into syringes here on Earth. The therapeutic compounds are preloaded into the multi-well plates here on Earth. We fly them up, and then the astronauts get paid the big bucks to take the syringes and inject them into the multi-well plates. At that point, they're simply placed into a plate reader, and that plate reader will monitor the amount of light that's being made by the cells continuously over a 24-hour period. So very little hands-on involved in this process, and something that, with the proper hardware, could be fully automated in the future.
Did you say you're going to look both at phase 1 and phase 2?
At this point, we're looking at compounds that have known effects, both those that have successfully been brought to market as well as those that failed during the development process.
Very good. Thank you.
All right. Thank you, Dan.
Thank you.
All right. Our next speaker is Dr. Torsten Nobert of the National Space Institute at the Technical University of Denmark and principal investigator for the Atmosphere-Space Interactions Monitor. He's going to talk to you about studying severe thunderstorms.
Thank you. ASIM is a payload that's developed by the European Space Agency to study severe thunderstorms. In particular, what we're interested in is gigantic lightning shooting up from thunderstorm clouds up to the edge of space. We will also be looking at flashes of high energy radiation that comes from thunderstorm clouds. It's called terrestrial gamma ray flashes. The things that we are looking for are really new, relatively newly discovered. Perhaps we've known them for like 15, 20 years. So ASIM will be the first time that we launch specially designed instruments to look for these things in space. And I think I have a video that can show you at least one of the animals that we're interested in. It's a lightning that shoots up from a thunderstorm below. You can see the lightning in the clouds. And then from the top of the thunderstorm, it shoots up to the edge of space, you could say, up to the ionosphere at 80 kilometers altitude. You can see the scale there on the left. This is not known for more than maybe 15 years. We call it a gigantic jet, and some of you might have heard about these flashes like red sprites and blue jets, and this is then a gigantic jet. Okay, so the instruments that we have are a big X and gamma ray monitor and a couple of cameras and photometers. And I think we have a video of these instruments as they are mounted on a pallet, which is being brought up by SpaceX, takes the pallet out. I'm not sure it's going this fast in real up there. It would be interesting if it was, but in any case, there you see it. So we are looking downwards. We are mounted on the outside of the ESA Columbus module, and then we're looking down. There's no astronaut, let's say, manipulation with these things. It's just looking down and taking images all the time of flashes of X and gamma rays, and of these new lightning phenomena. And so why are we interested in this? Well, of course, first we would like to understand what is it? And they have been observed for a while from mountaintops, for example, and there's been a few satellites also. So we know a lot about the physics, but we don't really know particularly well how, for example, terrestrial gamma ray flashes, how they're generated. So by this combination of instruments, we think we can take a step into understanding these animals further. Now, that's the first point. The second point is that up in the dilute atmosphere, everything happens more slow than down on the Earth with lightning. It's slower and it's larger. So what we are looking at in the upper atmosphere is actually lightning sub-processes, which are difficult to measure down on the Earth. Because lightning is so fast, it's gone before you're able to measure it very well. So what happens inside lightning, we don't really know that. But if you go to the upper atmosphere, we can see these sub-processes, and they happen slow, and our instruments can follow it. And then we are on the space station, which is like being on a balcony, viewing the whole scene, okay? And it flies all over the Earth, all the major thunderstorms, plus minus 51 degrees latitude. So we have this fantastic view of this upper atmospheric laboratory where we can measure fundamental lightning processes. So we think we learn a lot about lightning also. And the last step in the story I will tell you is that if we know more about lightning, we know more about what lightning does to the atmosphere. Ozone, NOx, trace gases that are important for our radiation balance and for climate. We'll know much better what lightning does and what role thunderstorms play in the atmosphere dynamics and in climate. So these were the words.
Thank you, Torsten. Do we have any questions for Torsten? Yes, over here. If you could just wait a second for a microphone.
Hi, Robert Perlman with CollectSpace.com. You mentioned that there's no astronaut involvement and it's just looking down on Earth. Is there something that triggers it to record, or what is it picking up?
Yes, exactly. They're running all the time, the instruments, they're running all the time. And whenever there's a flash, there's onboard software that captures those images with, those data that has a flash in it. Everything else is just overwritten, thrown away, but when the flash is there, it's put into memory and downlinked whenever there's opportunity to do it.
As a follow-up, are lightning flashes significant at that altitude? Are lightning flashes significantly bright enough to be distinguished from, like, let's say, aurora borealis or other phenomena, so that it's only lightning that you're picking up?
We will also measure aurora. It's interesting, your question, because it's actually auroral physicists that discovered these things. They were developing cameras to look for the aurora in the wintertime. In the summertime, test the camera, they see a red sprite, okay? So in the beginning, it was space scientists, auroral scientists, that were actually studying these things. And they don't know anything about thunderstorms, okay? So we had to bring in a whole new community to get to the physics of this, which was very interesting, I would say.
I think we did have one more question up front here. There's a microphone on the way.
Randy Siegel, WSTU Radio. What is the lifetime of the experiment that's going up on the station?
We are guaranteed 2 years. So if nothing else comes up to the platform and we function, we hope to get more than 2 years. But we are guaranteed 2 years. Yeah.
Thank you.
Yeah.
All right. Thank you very much, Torsten. Next, I'd like to introduce Rich Bowling. He is the Vice President for Corporate Advancement at TechShot, and he's going to talk about the multi-use variable G platform, which will serve as a new test bed to host experiments at Space Station. Rich?
Thank you, Marie.
You're welcome.
So this is a great week for the nearly 50 team members at TechShot. We've been working on the MVP for several years. We've designed and manufactured every single bit, every circuit board, every latch and component in-house. And to see it handed over to NASA,
to see the samples loaded in it and to see it finally launch will be great for the folks at TechShot. So TechShot, you might think of as a dry goods merchant for the 21st century. So while we typically are not the group that's doing the research in the machine itself, and you can see we've got a prototype of our MVP here with us today. We really are a company like those old dry goods merchants providing the picks and shovels that other folks are using to make new discoveries in the life and physical sciences. So to show you how it works, let's see a video of the system, which really consists of the payload locker that you see here on the platform, which is typically what you might know as a single mid-deck locker equivalent, about the size of a microwave oven. So the crew would remove the front door once the payload is on orbit, and then you can see the internal carousels. There are 2 carousels inside, and the sample modules fly up separately. The locker, the MVP locker, will stay on board the station permanently, and then only experiment modules will be inserted by the crew over time on different research campaigns. You can see on the upper and lower carousel an experiment module being installed on that, and then the crew puts the door back on. And then it's really automated, so we can control that from the ground at our Payload Operations Control Center in Greenville, Indiana, or at our office here at the Space Life Sciences Laboratory at the Kennedy Space Center. So anything that you can put inside one of those experiment modules, like you saw in the video, or what I have in my hand, each one is about 800 cc's of volume. So this experiment module could be used for plants, fish, cell experiments. We're doing a project with tissue chips from a team at MIT coming up this year. And you can also use it for things like fruit flies. And today we're so excited that the first user of our MVP payload is NASA. NASA is our very first customer. And so perhaps we could hear from the team from NASA about what they're going to do with the TechShot MVP.
Absolutely. Thanks, Rich. And we will have an opportunity for questions for Rich, but we do want to bring in first Dr. Sharmila Bhattacharya. She's a senior scientist at NASA's Ames Research Center to talk a little bit more about that.
Thank you, Marie.
You're welcome.
Thank you, Rich. Hello, everyone. So I'm going to tell you a little bit about the science that we're doing in this hardware that Rich described to you. And then I'll tell you how the hardware really helps us maximize the science output that we can get. So if we could show the first still, please. So this first experiment, as Rich mentioned, is using fruit flies or Drosophila. So what the experiment is about is that we're going to use the fruit fly as a surrogate host. There are a lot of similarities in the immune system between the fly and the human in the innate immune system. So we're looking at the effects of spaceflight on the immune system of this host model system. We're also flying up a bacteria, a pathogen. It's a pathogen for fruit flies, and then there's a related species of that pathogen that is pathogenic to humans as well. So what's interesting here is the mechanism by which there could be a change in that virulence or pathogenicity of the bacteria, coupled with how the host's immune system is going to respond, is of interest. And so some of this actually is a follow-up, leads up from work that has been done in the past. With fruit flies, we've shown, for example, that in space, the immune system shows a decrement. It shows some decrease in function, some changes in function. And some of those changes are often similar to changes that you've seen in astronauts. Similarly, other people have done a lot of work on the microbial arena, so with microbes or with bacteria and other pathogens, where they've shown that in the space environment, these pathogens may show a different physiology and may become more virulent or can grow faster or behave differently than they do on Earth. So for NASA, it's of interest to see what this combination of a host immune change and a pathogen change can do. And so here, what we're planning to do is to use the fly sort of as a surrogate because you can get large sample sizes, and I'll show that to you in a second with the hardware. where you can use this as a model system to test different kinds of pathogens and their changes as they're happening in space. You can process it and do this with high N number, so large statistical size of samples to look at the changes. So how does this hardware specifically really help with experiments such as this and other experiments in the future? For example, other people in the past on SpaceX missions have used flies to study cardiovascular systems or neurobehavior. So how does hardware like this help us going out and doing science in the future? Well, there are several very important factors, but I'm going to highlight 3 to begin with. One of them is, as I mentioned earlier, sample size. So we can get really good size populations. And the way this works is if you started, for example, there are food cylinders. Now this is obviously all closed, but there are food cylinders here which have the food that is very nutritious to the flies. The flies can then develop a generation in a chamber that goes down this way. And then they lay eggs, so you get generational separation. They lay that next generation of eggs into the food cylinder here, which then in space can automatically turn, so again, you don't need crew time for that, turns and you get another generation of adults developing in here. Those can then lay eggs here. Now, at any of these points, you can also take it out and fix them so that you can look at the status of health or gene expression or whatever you want while they're in space station. Or you can leave it like this and do multi-generational growth over actually several months to a year if you send up enough hardware. So the other good thing is these chambers are big enough. In addition to separating the generations, you get good population sizes. So we, in fact, with the TechShot team, loaded 50 flies for this first chamber, and for each of 12 units, so 600 flies is what we loaded this morning, right, Rich? Yeah. And then we expect, when we get it down in a month, we expect to get at least 100 times that. So that's the kind of, because they're going to reproduce. And what that brings about is we know that astronauts can go into space and then they come back, right? They're alive, etc. But we also know they undergo physiological changes. So we know there are important changes that happen to organisms, but they're often subtle. So when you have large population sizes, you can do the statistics and you can actually, and that's what you use model organisms, even on Earth, to do studies to get the statistics. The second really important feature of this hardware that will be very helpful is the centrifuge that Rich talked about. So the centrifuge, why is that important? Because it provides a 1G environment in space. So you have 3 sample sets. You have your microgravity sample set, you have a 1G sample set in space, and you have your 1G ground set. So that you can start differentiating, when you see a biological change, you can start differentiating and say, is it gravity that's affecting it? Or is it some other aspect of space? Is it radiation? Is it some other stressor of spaceflight? Also, the fact that you can separate these generations and you can fix the samples means you can distinguish the stress of landing or takeoff from being in space for a period of time, because you're actually able to fix your samples and then analyze them later because you've kind of frozen them in time in space. The other very important thing is, of course, it has all the bells and whistles. In fact, I always used to joke and call it that when we have this hardware, it will be the Cadillac of Drosophila hardware. But given that it's SpaceX, I have now changed it to say it's the Tesla of the Drosophila world. But the idea is that, so the other bell and whistle that is very useful is that it has temperature control. It has relative humidity is well controlled. So the environment is extremely well controlled in the habitat, which, as you know, for any living organism is a key factor. It also flows air from the cabin so that the oxygen can be diffused. The CO2 can be diffused. The oxygen comes in, and so the flies are in a healthy environment. And of course, it has cameras, so you can get behavioral data. It has lights, circadian lighting, and all of that. So, you know, I think this will be an extremely valuable piece of hardware to do science in the future. And I think with that, I'll stop.
Okay. Yep. So I think we have probably just enough time for not more than one question for each. So any questions for Rich or Sharmila? Okay.
There's one thing I should add about
Sure.
MVP, that we've talked about the 1G, the importance of the 1G control. But again, we can do fractional gravity as well. How much gravity is enough to be healthy? So we can reproduce the gravity of the moon and the gravity of Mars and anything else you want up to 2Gs from 0 to 2. And I think that'll be really helpful going forward with a number of different types of investigations.
All right. Thank you both. All right. With that, I will introduce our final speaker. Last but not least, we have Dr. Howard Levine. He's Chief Scientist in the Utilization and Life Science Office at NASA Kennedy Space Center to talk about continuing research on growing food in space.
Thank you. So tomorrow we're launching 7 new ways of growing plants in space, or 7 of these units. We've spent the last 2 years developing prototypes here at Kennedy Space Center for this new way. It's essentially using a plant cylinder within a reservoir. It uses capillary action to bring the liquid up into the plant reservoir where there's a substrate and slow-release fertilizer pellets. The seeds are up here. They germinate, they grow within the veggie unit. And it's replacing the current baseline nutrient delivery system, which is shown in the video. If I could have the video now. So this is veggie. There's 2 veggie units on the International Space Station. They use pillows. There's 6 pillows, each of which has seeds, and in this case, red romaine lettuce. We've had issues with some uniformity of growth of the plants, some problems with seed germination. So we've developed a system to mitigate those issues, reduce risk, and here you see some of the new ponds units being planted in the laboratory by some members of the science team. So my assistant, his name is Dave Reed. He's more than just an assistant. He's the project manager for TechShot, the same company that developed the centrifuge you just saw, and they partnered together with Tupperware Corporation to develop the flight-rated unit, which is here. So Tupperware, thank you. So Tupperware has used their advanced injection molding technology to develop this system. It's very slick. It's all kind of bayonet mounted, and we're just very excited. We think we're going to get incredible plant growth results, all passive, no electricity at all, and we're looking forward to the results. I have a few minutes, so I'll just say that on the launch tomorrow, there will be 7 of these that go up. Let's see, 3 of them will be like this, darkened, opaque. 3 of them will be clear with covers so that periodically the crew will be able to lift up the covers and see the progress of growth, root penetration into the reservoir. And then there will be another one which is completely clear that the crew will just use to video water distribution patterns within this unit, because that's our big uncertainty. Where is the water going within the reservoir? So in addition to this, on OA7, orbital ATK launch in, I believe, May, there'll be another 6 going up. And so the 6 that are going up tomorrow will be growing red romaine lettuce, and the 6 that go up in May will be growing mizuna. So these are both what we call pick-and-eat salad crops, things that the astronauts can harvest, wipe down with some sanitation wipes, and either consume directly or send back to Earth for scientific analysis. So with that, I think we can take questions.
All right. Any questions for Howard? Yes, right here. There's a microphone coming.
Randy Siegel, WSTU Radio. How much of the growth that's happening with lettuce on board is being consumed, and do they have to first send them down to get approval to consume them?
So we went through a very elaborate approval process with the flight surgeons, and we collected data on the microflora, the microbial community that colonizes the surface of the plants, as well as the And we were able to convince the flight safety folks, the flight surgeons, that with this wipe down procedure, it's sufficiently clean for the crew to consume. And they consume, in general, in most of our experiments, about half of the tissue. And we do experiments where, you know, at the beginning we grew, let's say, lettuce for 30 days and we harvested it all, but we've moved past that. So we're doing something that we call cut and come again, where we'll harvest some of the plants, you know, the outer leaves, and let the inner actively growing portion continue, so we can get 2, 3, maybe even 4 harvests from individual plants.
Okay, and I see another question over on the side here.
Hi, Robert Perlman with Collect Space. Given the very recognizable brand name of Tupperware, is there any one-to-one comparables of what an average person might find in their kitchen from Tupperware to what they actually built for the experiment?
Well, I would say Tupperware uses a very sophisticated injection molding procedure. And they use that same procedure for the fabrication of these flight units. Actually, 5 different molds are used. And actually, there's, I believe, a few more pictures, which I think if we have time we could show. So here's some tomatoes that are grown in some of these units here at Kennedy Space Center. Next slide, please. Here's the team loading up the ponds units. Next slide, please. And this, so you can see that there's 6 of these units that will fit into each veggie unit, which I think many of you are familiar with, as implemented on the International Space Station. So it's pretty much a one-to-one correspondence with the currently existing plant pillows.
Okay. I think we also had a question on social media.
So we have a question on social media. They would like to know, is part of the experiment to bring lettuce back to Earth that is grown in space? And how do you compare it to the lettuce that's grown on Earth? Does it vary in structure and taste?
So yes, about half of the harvested tissue in general in most experiments is brought back to Earth for analysis. That's for nutrient content, things like isoflavonoids, vitamin content, some of these micronutrients that don't survive as well when you do long duration food storage for, let's say, deep space missions, and also analysis of the bacterial community composition on the surfaces. And it's pretty much comparable in growth rate to what we see on Earth. 30-day grow outs for lettuce is usually what you'll get for a nice head.
Okay, we have time for one more question. I know you had one over here.
In terms of weight for these units, are they lighter or heavier than the veggie units that are currently up there?
They're about the same. But one big thing is that these are reusable. So the pillows that we currently have up there, it's a one-time use and then they get tossed into a progress, they burn up. These can be one-time use, or we can bring them back, and in this experiment we're bringing them back. So we hope to refurbish them and reuse them. And in the future you could, we believe, refurbish them on the International Space Station and reuse them.
Thank you so much, Howard. Thank you to you and to all of our presenters. And thank you all for joining us on this Sunday afternoon. That'll do it for us here. For more information about what's on board, you can always go to www.nasa.gov.